Alright, in this video we're going to discuss DNA replication. Alright? So, before we can really get into DNA replication, though, we need to visit DNA structure. Alright? So, you should be pretty familiar with some of the basics associated with DNA, but let's just look at it. Alright, so if we have a single strand, we normally orient it, something, so we'll go at 5' at the top, 3' at the bottom. Alright, it always has a complementary strand, right? So we put a second one here, and I'm going to make this one 3' at the top, 5' at the bottom. So we refer to that running in opposite directions, right? 5' to 3' top to bottom, 3' to 5' top to bottom. We call that anti-parallel. Alright, and that's going to become very important when we look at the actual DNA replication, right? That we can only synthesize DNA 5 to 3, and we can only read DNA 3 to 5. So that's going to be extremely important. Alright, we have base pairing, right? So if we look at our nucleotide bases, alright, so we have base pairing rules. So if we put an A here, we of course have a T here, and if we put a C here, we have a G here. If we have a G here, we have a C, and so on. Alright, so we've got base pairing rules. Right, we've got this anti-parallel nature. We are going to read, alright, DNA, so the template. So we're going to read the template. Three to 5. Okay, we are going to synthesize DNA 5' to 3. Okay, so those are our basic rules. All right. The other thing that we want to keep in mind about the DNA structure is that when we make it, we replicate it semi-conservatively. All right. So we have semi-conservative replication. Okay, so what that means is if I start out and I have a double-stranded DNA molecule like so, alright, I'm going to replicate this into 2 DNA molecules, right? And they will each be 1/2 of the old, and we'll just make our new in this pink color here. Alright, and so this would be the new, right? This would be the old, and so it's semi-conservative. Alright, when we're looking at synthesizing DNA, we would have called this one would be the template strand, right? That's the one we use to synthesize the new one, and over here this is my other template. Okay, and so you want to keep those things in mind. Alright, so let's go ahead and look at replication. Alright, so we have what is called a replication fork or a replication bubble. Alright, okay, so this one to the right is heading in this direction, the one on the left is heading in that direction. We have an enzyme here called helicase, alright, which, as you know, unwinds the double helix. Alright, we have an enzyme called topoisomerase. Alright, which, as we unzip the double-stranded helix, double helix with the helicase enzyme, it creates a lot of tension out here. Alright, and the topoisomerase relieves that by creating single-stranded nicks in the DNA to allow those twists that are being created to basically be relieved. Alright, so the other thing that we want to look at here, all right, and so let me get rid of a little of this. Okay. The other thing we want to keep in mind is the orientation of things. So what I'm going to do is let's just look at this one over here on this side. So if I make this my 5 prime and this little starting point here 3 prime, okay, and I make this one has to be anti-parallel, so this is 5 prime here and 3 prime here, right? So we are reading the DNA, okay, so we are going to read it 3 to 5. So we can read this bottom strand in this direction. We can read this top strand in this direction, right? That's our 3 to 5. So those red lines, that's our reading, okay? Now we can synthesize 5 prime to 3, all right? So if we think about synthesis, all right, 5 prime to 3 prime, this top one here, it can just move in this direction, right? The bottom one, however, all right, if we unzip initially to this point right here, all right, I can build a primer and I can synthesize in this way, in this direction, okay? Now, if I unzip it further, right, so helicase and topoisomerase are doing their job, I can put in a second primer and I can synthesize in this direction, right? This is still sticking with my rules, 5 prime, 3 prime, right? 5 prime, 3 prime, okay? So we call this our leading and this our lagging strand, okay? All right, so let's just look at a replication fork, and we'll do it very similar to the way your book does it, all right? So if I'm going to make my replication fork, I'm going to go like so on the top and like so on the bottom. Okay, I don't know why that. Okay, let's label this 3, this 5, make it anti-parallel, 3 and 5. Okay, so we have a couple of enzymes out front, which we've already talked about, right? So we've got helicase here, and we have topoisomerase here. Okay, now the first thing that's going to happen is, and let's start with the leading strand. And so if we're reading the top strand 3 to 5 in this direction, right, for my leading, okay, so what we want to do then is we need a primer, okay, and this is RNA, okay, and RNA is, this is the enzyme involved in making this is called primase, okay, it's a it's an RNA polymerase. It builds our primer, which provides a 3' hydroxyl, all right, provides this 3' hydroxyl that is needed by DNA polymerase 3. All right, remember DNA polymerase 3 needs a nucleotide to add its nucleotides to. Okay, so the orientation then of our primer, right, is 5 to 3 in terms of how it is synthesized. Okay, so we have a 5 prime here and a 3 prime here. Okay, now the DNA is synthesized 5 prime to 3 prime, and since this is our leading strand, our DNA is going to be synthesized in this direction here, okay, like so, and out here this would be my DNA polymerase 3. Okay, now that's the leading strand, and so that just follows this replication fork, you know, in the direction it's heading. Now we have to also though remember that DNA can only be synthesized 5 prime to 3 prime. All right, so if we look at this bottom strand, this bottom template, all right, it is being read, all right, in this direction, right? So it's being read in that direction. So initially, let's say we open up the DNA to right about here, all right? So we put in a primer, all right? Then we would synthesize the DNA, okay? Then if we open up this black DNA, right? These black lines that resemble our initial template DNA, we open it up, say, to about here. We can put in a second primer, all right? And then DNA polymerase will, of course, come in, and it will go until it runs into that first primer, all right? If we open it up yet further, all right, put in yet another primer, and we're just getting started then on this last one. Okay? So this one was first, right? This one is second, and this one is third, and eventually there would be a fourth and a fifth, and so on. Okay? As the way it works. Okay. So now that we're starting to build these fragments of DNA for our lagging strand, and we call these things Okazaki fragments. Okay? So we're building these Okazaki fragments. The problem that the cell has, though, is that these Okazaki fragments they're in pieces, and we have to stitch them together, right? We have to get them together, and there's RNA in between them all. So what happens then is, all right, there is an enzyme, DNA polymerase 1. Okay, so DNA polymerase 1, all right, it is going to come in right here, all right, I'll draw it, all right, and what it will do is it will remove the primer, okay. Of course, I removed it too, but we'll get it back, all right, and it is going to extend this fragment almost to where basically that needs to be bonded to our initial fragment. So it's taking fragment 2 and extending it so that it runs up against fragment 1, and it's removing the primer of fragment 1. Okay, so it's going to remove the primer from fragment 1, and it's going to extend fragment 2. Okay, all right, so that's what DNA polymerase 1 would do. Okay, so that's it there. Now, once DNA polymerase 1 is done, all right, what's going to happen is an enzyme called ligase is going to come in. All right, and so I'll make ligase green. All right, and so the enzyme ligase. And what ligase does, it's a G, all right, what ligase does is ligase is going to make this final bond right here and make this one continuous fragment. Okay, so if I have ligase making this final bond, and I'll make this little arrow here, all right, between my fragments, all right, we can put our other enzymes on here. Okay, so we can put DNA polymerase 1, all right here, chewing this, getting ready to chew that away, if we extend this one to here. Okay, so now this is going to come in, all right, and we call that activity of the DNA polymerase when it's removing RNA. Okay, we call that activity ribonuclease activity. Okay, or you can also call it an exonuclease because it is excising or removing the bases, all right, particularly from the end. Okay, and so that is the basics of DNA replication. All right, so we have our leading, our lagging, and of course we have this other side of the bubble. All right, so what I suggest you try to do is take a DNA strand, right, is just draw yourself one of these, okay, label the top, label the bottom, you know, in terms of their, 3 to 5, right, make sure that they are anti-parallel. right? You need to identify the leading and the lagging, right? Leading and lagging, and then just build it, okay? Just as we just did, and I really do suggest you work that through on your own. The other thing is know the names of the enzymes and their functions, right? So names of the enzyme and function, okay? And I'll put functions, because particularly for DNA polymerase 1, it has multiple functions, okay?